So while they get my slides up, I'll just say good morning to everybody, and thank you to Amber and Mark for the invitation to present here. So for those of you I don't know, my name is Katie Helbig. I'm a genetic counselor at the Children's Hospital of Philadelphia. I provide genetic counseling to families in our epilepsy genetics clinic, and I'm also the co-director of our epilepsy neurogenetics program. So we care for many children with SLC6A1 at our hospital and in our program. And I wanted to just kind of set the stage for those of you who haven't seen any kids who have this condition to let you know what the clinical features are. Great, so we've got my slides up there. So I'm going to focus on what is known in the scientific literature. I think my talk will be complemented nicely by Dr. Bain, who's presenting some data from the family registry. But this is based on what we know about in the clinical literature. So I'm not going to spend too much time on the functional aspects of SLC6A1 because I think this will be covered nicely by other speakers later. But SLC6A1 encodes the instructions for a protein that's called GAT1, and this is a GABA transporter. So GABA is the main inhibitory neurotransmitter in our brain, and the function of GAT1 or SLC6A1 is to remove GABA from the synapse. So it's kind of like a little suction that sucks the GABA up and removes it from the synaptic cleft. So I'm calling this SLC6A1 deficiency disorder. I think that Dr. Demarest may have called it that last year, and I kind of like that name, but I think maybe we can decide as a group what we want to call it. It was first implicated, the SLC6A1 gene was first implicated in disease only 4 years ago, and this was by Gemma Carville and Heather Mefford, who identified 6 individuals who had an epilepsy with myoclonic atonic seizures, which used to be called MAE, or DOSA syndrome, who had pathogenic variants in this gene. So based on that initial cohort, it was estimated that perhaps up to 4% of kids with this EMAS phenotype actually have SLC6A1 deficiency disorder. That was followed up 2 years ago now, almost, by a study from the Danish group, and Katrine Johansen is here today. And this was a larger cohort of patients with this condition. So there were 34 individuals in this cohort, and that really defined the phenotypic spectrum a little bit better. And this is where most of our knowledge about the clinical presentations comes from, at least in the scientific literature. Did I skip a slide? Oops. So as of today, there are at least 50 individuals published in the literature. There are probably more who have SLC6A1 deficiency disorder. There are at least 70 unique pathogenic variants. There are probably many more, but this is what's available to us in public databases. And the phenotypic spectrum of the condition has really expanded beyond the initial publication to include much more than this epilepsy with myoclonic atonic seizures. So what I'd like to do over my next few slides is just present what this condition looks like based on our knowledge today. So as Amber mentioned, epilepsy is one of the main features of this condition. It's seen in over 80% of individuals who have it. The mean onset is around age 2, but it can really span the spectrum. So some kids present earlier in infancy. Some children don't develop seizures until later in childhood, but on average, usually kids with this condition develop seizures around the age of 2. 65% of them are able to become seizure-free, but that means that 35% of them aren't. So seizures are an ongoing problem for many children with this condition. In terms of syndrome diagnoses, most of them fall into this MAE category, the epilepsy with myoclonic atonic seizures, but a third of them have different presentations as well, including generalized epilepsies, childhood absence epilepsy. Some patients have a more severe developmental and epileptic encephalopathy. And then there are some patients reported who have a focal epilepsy, but largely this is a generalized epilepsy syndrome. In terms of the common seizure types we see, typical absence seizures are the most common. So both typical and atypical absence seizures, which are these staring spells that Amber mentioned before. And many children also have atonic seizures. So these are sometimes called drop attacks, where children quickly lose tone and fall to the ground, and they can be very dangerous. Myoclonic seizures are common. Some kids have a combination of these myoclonic and atonic seizures, where the atonic component will be preceded by a big jerk. And then other less common seizure types include focal seizures, sometimes eyelid myoclonia, and tonic seizures. But you can see that generalized seizures really predominate. The developmental aspect is significant as well. So 90% of individuals with this condition have some level of cognitive impairment or developmental delay. Usually that falls in the mild to moderate range, but some children are more severely affected. But what's interesting is that a small slice of children have typical development. So although most children do experience developmental delays, that's not seen in everyone. And the other neurological features are significant as well. So if you talk to families, and I encourage you all to speak with some families who are here today, seizures are a problem, but the other neurological features can be equally difficult or debilitating for the kids. So behavioral difficulties we see in some kids, including aggression, ADHD. The movement disorder component is significant, as Amber mentioned. So this typically involves ataxia or tremor or problems coordinating their movements. And features of autism spectrum disorder are seen in about a quarter, at least, based on what we know of now. So these are all, I think, equally important to the families of these children, that we can somehow find a way to address these conditions as well. So in terms of the genetic spectrum, as I mentioned, there are at least 70 pathogenic variants. There are probably many more reported in the literature. There are some recurrent variants. This is the most common one, this alanine 288 valine. It's seen recurrently for some reason, and that's the most common one, but really there are several novel variants that are unique to each individual child as well. And this is just a schematic of what the protein looks like. So this is the cell membrane. These regions are the transmembrane regions, and I just plotted out missense variants versus truncating variants. You can see that they're spread out across the protein, so they don't seem to cluster in any one particular spot. And this is also consistent with a haploinsufficiency mechanism, since we see it across the whole gene. So most of the reported variants are missense variants, so this is a substitution of one single amino acid, but you can see about half of them are loss of function or truncating variants that lead to likely haploinsufficiency. So 30% of affected individuals have a loss of function variant, including a frame shift or a nonsense variant. Splice site variants are seen in about 10%, and a small proportion have a deletion of a single amino acid or an in-frame deletion that doesn't shift the reading frame. So the mutational spectrum is pretty broad, and we see all types of variants in this condition. And the inheritance patterns are important to know as well. So about 3/4 of these genetic variants are de novo, which means that they occur spontaneously or happen new in the child. So it's genetic, but it's not inherited. But 25% of children, the variant is inherited. So either from an affected parent who may have various features of the condition, or importantly from a mosaic parent. So about 10% of reported cases were inherited from an unaffected carrier parent who's mosaic, which means that they only carry the pathogenic variant in a subset of their cells. And this is important to know for recurrence risk counseling, because typically when we see a de novo condition, this implies that this is a new event that happened in the affected child. But if a parent is a mosaic carrier, then the risk of recurrence goes up to 50%. And we have seen this in our center. We've seen mosaic carrier parents with multiple affected children, and this is certainly supported in the literature. So if you ever see patients clinically, I think it's important, if it looks apparently de novo, there is still some residual chance that a parent could have additional affected children if they're mosaic. So in terms of treatment, there really are no precision treatments yet, which is why we're here. We're hoping to change that. Like I said, about 65% become seizure-free, but there's over a third who have ongoing seizures. And the developmental concerns seem to be unrelated to seizure control. So usually children will have developmental delays before seizure onset, and even if the seizures are well controlled, that doesn't really correlate with developmental outcomes. So there is more going on in the developmental pathway than just ongoing seizures. There's some suggestion that Depakote or sodium valproate may be effective, but this is not really a precision treatment, because this is the first-line treatment for kids with myoclonic atonic epilepsy, or EMAS anyway. So it can be helpful, but it doesn't really necessarily target the underlying genetic pathway. There's some suggestion, there was one published patient who did well on the ketogenic diet, so I think that remains to be explored whether the ketogenic diet may be helpful as well, but that's largely anecdotal at this point. So how common is this condition? Amber touched on it before, that the prevalence is estimated to be about 1 in 38,000, but this is one of the more common genetic causes of epilepsy and developmental delays. So in an unselected cohort of individuals who underwent epilepsy panel testing, it accounted for 2% of all diagnoses, which is a pretty heavy hitter in the world of epilepsy genetics, where there are over 100 different genetic causes. So to see 2% is quite high. The initial paper suggested that 4% of all children with epilepsy with myoclonic atonic seizures have SLC6A1 deficiency disorder. A cohort of adults, which is a largely untested population. So there are probably many adults who have this condition who have not had the proper test to diagnose it. And a recent study indicates that maybe 1 to 2% of adults with epilepsy have SLC6A1 deficiency disorder as well. So I think it's important that we reach our adult neurology colleagues as well. And then I think this is a really nice study that came out of Scotland, which was a population-based prospective study where the authors tested all children under the age of 36 months who presented with new onset seizures. So this really gives us a better sense of how common this is on a population scale. And this was the fifth most common genetic diagnosis in this prospective population-based cohort. And it was seen in about 1% of all kids in this cohort. And there were 8 kids with epilepsy with myoclonic atonic seizures, and one of them had SLC6A1 deficiency disorder. So it certainly is a common genetic cause for this epilepsy syndrome. So it's common. In terms of rare disease, this is a less rare condition, so I think it definitely deserves our attention. Genotype-phenotype correlations is something that we're always interested in, and it's something that families ask a lot about when they're getting the diagnosis. Can we predict anything about the outcome based on the underlying genetic variant? The answer to this is we don't know. It hasn't really been explored in the scientific literature. We think probably not, at least based on our own anecdotal experience. We can see vastly different clinical presentations in children who have the same exact genetic variant. So the genetics is not a crystal ball. It's an explanation, but it doesn't really give us much information about potential long-term developmental outcomes. But I think that this should be explored. So there have been no systematic studies of genotype-phenotype correlation, and this may be worth looking into. So to summarize, SLC6A1 deficiency is associated with childhood onset generalized epilepsy in over 80% of individuals, usually starting around the age of 2, but it can span from infancy until later childhood. The most common seizure types are generalized seizure types, usually absence seizures or atonic seizures. Seizures can often be well controlled, either with valproic acid or maybe the ketogenic diet, but there are a significant number of kids who have ongoing seizures. The developmental delays seem to be independent of the epilepsy. They're seen in over 90% of kids, and they're often apparent before seizure onset. And most of the developmental impairment we see is in the mild to moderate range, but there can be quite a broad spectrum, and there certainly are children who are more severely affected. The other neurological comorbidities are important, so we see ataxia and coordination difficulties in about 30%. Autism spectrum disorders are significant as well, up in about a quarter of all kids, and there are no clear genotype-phenotype correlations. So this is what we know about SLC6A1 based on the literature. I think that Dr. Bain's presentation will nicely complement mine. And I just wanted to give a shout out and thanks to my team at CHOP, the Epilepsy Neurogenetics team, who do a fantastic job of caring for our kids with SLC6A1 deficiency disorder. So I think we'll have questions after Dr. Bain's talk, and I guess she can come up here and present. Thank you all very much for your attention.